what is DNA and what does it do? Now the DNA stands for deoxyribonucleic acid. It is deoxyribonucleic acid, and hence the name DNA. Now the DNA is also called the master molecule of our body. It is a very important molecule of our body because in the DNA, the whole instruction manual of our body, the way our body grows, the color of our eyes, the type of the skin that we would have, the color of our skin, the kind of hair, and many other characteristics. There are about 40,000 characteristics in our body. They are all controlled by the DNA. So it is called the instruction manual of our body, and hence the master molecule. And during reproduction, this instruction manual, that's a DNA, passes from the parent to the children or to the offsprings. This happens during reproduction, and that's the reason why the offsprings look very much like that of the parent. There is a great similarity because it's the same DNA that passes from the parent to the offspring during reproduction. So the instruction manual of the parent passes on to the offspring, and hence the offspring look similar to the parent. So what does this DNA look like and where is it present? We have the cell, which is a basic unit of living organisms, and inside the cell we have the nucleus. Within the nucleus we have a network. There are other things in the nucleus, but there is this network, and this network is called the chromatin network. And the DNA is present in this chromatin network. Remember, the DNA are long molecules, very long molecules, and they are present in the chromatin network. This is for a normal cell. But in the case of a dividing cell, when the cell is about to divide, this chromatin network arranges itself into rod-shaped structures, and these are called chromosomes. This happens only in the dividing cell. Rod-shaped structures, and these structures are called the chromosomes. So here in the picture we can see the cell, and in the cell is a nucleus, and inside the nucleus is a chromatin network. This chromatin network then develops into chromosomes. These are chromosomes, rod-shaped structures called chromosomes. And when you open up the chromosome, when you amplify it, you get the DNA. So this is the DNA. This is an amplified form of the DNA. It looks like a twisted ladder. We can see, I'll put another color here. This is the DNA, which carries all the information. However, this part, the next part of the picture, is the replication of the DNA. How the DNA replicates or reproduces. So now we know what the DNA looks like. It looks like a ladder, a twisted ladder. It's called a master molecule because it carries the instruction manual of the body. So this is the picture of a DNA molecule, a long molecule. Now what are genes? Genes are functional units of the DNA. So these genes are little parts of the DNA. Like for example here, this could be one gene, and this one gene could be responsible for the color of your eye. It's an instruction manual for what the color of your eye should be. Or there could be another gene which is responsible for the type of hair that you have. It would carry the instructions of making the protein which will decide the type of hair you have, curly hair, or you have straight hair, you have dry hair. There could be another gene that is responsible for the color of your skin. There can also be genes responsible for the condition of your heart. So in this way we have many genes, and each of these segments of DNA, they're basically segments of DNA, are genes, and they control our traits, they control our characteristics, because they control the making of proteins, and hence they control our traits like tallness or short. Not only in humans, also in plants. Tall plant or a short plant, like you will see in the pea, you have the round pea, you have the wrinkled pea. All these are controlled by the genes present in the plant. So earlier we have seen we have this DNA which is present as a network, which is called the chromatin network. And this chromatin network, just before a dividing cell, this chromatin network arranges itself into rod-shaped chromosomes. Now in the human cell, we have 46 such chromosomes. Each human cell has got 46 chromosomes. We humans reproduce by sexual reproduction. They reproduce sexually. And in sexual reproductions, we have the male gamete, which is the sperm. We have learned this in the earlier chapter. And we have the female gamete, which is the egg, fuse. Now remember, there are 43 chromosomes here, 46 chromosomes, and here too there are 46 chromosomes. And when they fuse, we get the offspring. Now this would be 46 plus 46, 92 chromosomes. Now definitely this offspring with 96 chromosomes will not resemble the parent. Why will it not resemble the parent? Because the parent has got 46 chromosomes. And the offspring has got 92 chromosomes. So this cannot happen. This does not happen. This cannot happen because the number of chromosomes doubles. So actually what happens in humans and in other sexual reproduction, the normal cells have got 46 and these will have 46. Now this cell first breaks up into two smaller cells, each carrying half the number of chromosomes, 23 and 23. This normal cell with 46 will break up to form two smaller cells having half the number of chromosomes, that is 23 and 23. These are called the gametes. These cells are called the gametes, and it is the gametes then fuse to form the offspring. So this gamete and this gamete will fuse now, 23 plus 23, and you'll get 46 again. Maybe this gamete and this gamete will fuse, 23 plus 23, and you get 46 again. So this is an important process where the number of chromosomes are halved before reproduction. And this halving or making the chromosomes half, the process is called meiosis. These cells are called diploid. The normal cells are called diploid, or we call them like 2X chromosomes. And here these are called haploid. The gametes are called haploid, and they just have X number of chromosomes. So to just do it up again. So you have the 2X chromosomes, which will break up into X and X. These are the gametes. This is the cell, normal cell. Here again, the 2X has to break up into X and X. So this is haploid, one set of chromosomes, and these are diploid, two sets of chromosomes. And then the sexual reproduction can take place, X and X to give you 2X again, X and X to give you 2X again. So this point is important. I was trying to make this point clear. The need for 2X to become X. Now we'll learn the monohybrid cross. Before we do the monohybrid cross, let's just revise this fact that we have the gametes, and during sexual reproduction, the male gamete with X chromosome, the female gamete with X chromosome fuse to form the offsprings. Now the offspring has got X and X, two X chromosomes. So the point I try to make here is we all have two sets of genes, one from the father and one from the mother. Even plants also have two sets of genes. One gene will come from the male and one gene comes from the female. So keep this in mind that every cell has got two sets of chromosomes. One set of chromosomes coming from the father or from the male and one set of chromosome coming from the female. So one comes from the male and one set of chromosomes comes from the female. And however, when this organism then reproduces and forms gamete, it forms then the 2X then becomes again X and X when this offspring is ready to reproduce. In the monohybrid cross, Mendel took the characteristics of height. So when you talk about height, you can have two contrasting characteristics, tall and short. So in the first case, he took a pure tall plant. What you mean by pure tall? It had both the genes of tall. And then he took a pure short plant. What you mean by pure short? Two genes. Remember, we have two genes for every characteristic. One from the father and one from the mother. One from the male, one from the female. So this had two genes and both the genes were short. So we have the pure tall and he took the pure short and he crossed it. Now before he crosses, this is a cell. Now this cell has to divide into two gametes. The 2X has to become X. So this were the gametes. These two have to divide into gametes. Like we seen earlier, the 2X has to become X and X. So here we have the male gametes, maybe it's a male gametes, and here we have the female gametes. Now it's time for the gametes to cross. Now let's see the possibilities. We could have taken up with first this gamete would now cross with this gamete, and hence the combination we get will be tall and this tall. Now the same gamete would cross with this gamete, and now we would get this with this gamete here. It would be tall and with this short. Next, it will be could be this gamete will now cross with both these. So you have these two where it will be tall and this short. And last, we would have again the same gamete now could cross with the second one, and now we would have tall and this was called the F1 generation. And in the F1 generation, he found that all the plants were tall. Although it had this short gene, but all the plants looked tall. This clearly went to show that it was a tall gene was dominant. This T was dominant and it expressed itself. The short gene was unable to express itself because there was a tall gene. So in the F1 generation, all were tall. So the conclusion you can derive from here that the tall is dominant, is a dominant gene. And the small t could not express itself. The short could not express. The plants did not become short, could not express because of the presence of the dominant gene, due to the presence of this capital dominant gene. So we had tall here, this also was tall, and this was tall, and all were tall in the F1 generation, all tall. Then next Mendel did, he crossed the F1 generation. So the F1 generation we know both had this tall, and there was this short. So this is the cells of the F1 generation, and he crossed them. Now for crossing, first we have to find the gametes, right? We have to do the gametes, 2 gametes here. So the gametes in this case would be, one tall would come here, and there would be short here. The 2x become XX. And here too, the cell would now divide into 2. Next cell also would be 1T, and there would be another T here. So remember, these are the gametes. Now we're going to try and see what's going to happen in the F2 generation. So we have 4 gametes here, and to make things a little clear, we make it in a tabular form. So here we have one gamete, one pair of gametes. These are from here. And then we'll have the other pair of gametes. And now we will cross them. The first, this T with this T will give you T and T. Next we take this T now with this gamete, and we'll get T and this small t. Next we have this T crossed with this gamete. This we have to put in with this, and then we'll have T and T, and in the last we have this T with this T, and that would give us TT. Let us see what are the characteristics. In the first case we have T and T, and this would be tall. Here we have capital T, small t. This plant would also be tall. This next one will also be tall. And last, here we will have short, because both the genes are of recessive, they'll be short. So in this case we have 3 tall and 1 short. So the ratio is 3:1. 3 tall, 1 short. Let's go over the monohybrid cross again. So we had pure tall, this is pure short. Then we got the gametes. The gametes was tall, and this was tall. The gametes of these were the gametes. So 2x became x. And now on fusion, this with this, we got tall. Then this with this, tall. Now we have to do this with this. Oh no, with this now. We got tall. And then we have this with this. So 4 possibilities. This was the F1 generation. Now then we took the 2 F1 generations, tall, this, and we crossed. But before we cross, we have to make our gametes. So gametes was capital T, small t, and this gametes were capital T, small t. So we can see here are the gametes. This is the male maybe, and this is the female. And when you crossed, you got first here capital T, capital T. Here when you cross this T with this T, you got here TT. Next to cross this T with this T to give, then this T with this to give you, and last this with this to give you. Now this is important, the genotype. You had one tall, have you seen they had this one tall. Then we had two of them which are capital T, small t here, capital T, small t. There were two of them. And then there was just one that was small t, small t. So this was the genotype ratio. 1 here, 1 is to 2 is to 2. That means 1 is to 2 is to 1. That is TT, 2 of T small t and T of T. Now when we go to the phenotype ratio are the features, the external features. In this we had tall, the yellow were tall, this was tall, and this was short. So we have 3 tall, and 1 was a dwarf. So the ratio was 3:1. Tall and dwarf or short. Remember, this is the phenotype ratio, the features. And this was the genotype ratio of genes, the type of genes. We'll take another example of a monohybrid cross where you have a yellow seed, pure yellow, both capital, and a pure green seed. Green is small y, small y. So here first these are the cells. We need to get the gametes. These are the gametes. Now these gametes, I will put it this way, so otherwise it gets a little confusing. These are the gametes. Now the gametes will fuse. And for fusion, we have this gamete with this. So this will give us capital Y, small y. Then this with the same, with this one. So now you have capital Y, small y. And then we'll have this with this, capital Y, small y, and this with this, capital Y, small y. And you'll expect all of them to be yellow because the yellow is dominant. This also yellow. And here they've given it very nicely. The way we have done it in this way, they've done in tabular form. They put the 2 genes here, Y and Y, and these 2 genes, Y and Y, and on the other side they have written the small y and small y, and it's written here, see, small y, and they have crossed it. Here you got yellow, 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 and yellow. This is the F1 generation. All were yellow because yellow is dominant. In the next step, we cross the F1 generation. Now, if you recollect, the F1 generation has capital yellow like this. These were the 2 of the F1 generation, 2 individuals of the F1 generation. Their gametes would be capital, small, this would be the gametes, and here this would be the gametes. So in this case they have put them here. This Y and this small y has been written here. They're heterozygous because they've got different genes, and this Y of the female has been put here, and then you cross. In the first case, you take this Y with this Y, you get Y, Y which is yellow, Then you take this Y with this Y and you get Y and small y, which is also again yellow. Then you take this Y with this Y and here you get Y and small y. And you get this small y and this small y here. And that will be green. So here you had y yellow, y small y yellow, y small y yellow, and here both were small, so this would be green. So we have three yellow and 1 green. So ratio 3:1. This is the phenotype ratio, the features. But if you want to see what is the genotype ratio, here we have 3 kinds. We have capital Y, some gene capital Y, small y, and we have small y, small y. This capital Y, there are only 1. Small y is this capital Y, small y, 2, and the small y is 1. So the genotype ratio is 1:2:1. Here are some more examples, different kinds of examples. So here is a cross between TT and TT, means both pure. So it is a cross between this T, and this is what we have done. The gametes were T and T, and these gametes were T and T, right? So here is a cross between these two. In the second, of course, the characteristics here will be tall, 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 and tall. Now in the second one, it is a cross between capital T, small t, capital T, small t. So here one individual has got this makeup and the other individual has got this makeup. So the gametes in this case will be T and T. Remember, they become half. And the gametes in this case will be T and T. And here's a cross. This is the 2 is a cross of these two individuals. Here again, this will be tall. This will be tall. Here will be tall, but this will be short, because there is no dominant gene, there is no capital T here, so it will be short. Here's another cross between a pure, this is a pure TT, with a mixed, that is capital T, small t. So here we would have the gametes in this case would be capital T, which they have put, and these gametes would be T and T, and they put the gametes here. This would be the gametes here. When you do this cross, this will be tall, here will be tall, this also tall and tall, all tall, because all have this dominant gene. At least one capital T it has got. So all will be tall. Next is a cross between two recessive. This is short, two pure short, and this also is pure short. So the gametes here are T and T, and these gametes here are T and T. And if you look at this, this will be short, this will be short, all will be short, because there is no tall gene at all. Here both the parents are recessive, so all are short. You can just go through these examples to understand the concept better.